Welcome everyone, my name is Katharina Brazda and I'm currently a PhD student at the University of Vienna. In this small lecture series, I will talk about modeling biological membranes, like the red blood cell or the multiphase vesicle I've illustrated here. And the aim is to show you how their configurations can be described with tools of geometric measure theory, in particular curvature varifold. The lecture consists of two videos. In the first part, I will give you basic facts about the physics of biomembranes, and then I will explain the Canham-Helfrich variational model for the shapes, and finally present its generalization to multiphase membranes. The second video will start with a brief and hopefully gentle introduction to the theory of varifolds, specifically curvature varifolds, and then, using these tools, I will prove existence of minimizers for the multiphase Canham-Helfrich energy. Let me mention that this result is a joint work with Luca Lusardi from Turin and Ulisse Stefanelli from Vienna. So let's get started. What you can see here is a cartoon of an animal cell, and first I want to draw your attention to the variety of shapes and structures you can see. These are mostly made of biological membranes, most prominently the plasma membrane separating the cell from its environment. Also, the boundaries of the organelles and of the nucleus all consist of biomembranes. In particular, cell membrane material must be able to undergo dynamic morphology changes, like in exocytosis processes or cell division. So it is stable and flexible at the same time. If one takes a closer look, one discovers that biological membranes are a fluid mosaic. The basic building blocks are phospholipid molecules, which you can see here. But a cell membrane generally also contains other molecules like cholesterol, embedded proteins, and has attached sugars. Phospholipids have a hydrophilic head but hydrophobic tails, so they will spontaneously aggregate to form bilayers when put in an aqueous solution. And these bilayers will form closed surfaces, the so-called vesicles, without any free edges like this. The individual molecules like to be in a fixed distance to each other due to electrochemical forces of van der Waals type. And therefore, since the number of molecules is constant, the vesicle will have fixed area. Moreover, due to the osmotic pressure equilibrium between inside and outside fluids, also the volume enclosed by the membrane is fixed. So, from a viewpoint of continuum mechanics, biomembranes behave like two-dimensional viscous fluids with an elastic response to bending forces. For me, it is this combination of fluid and solid mechanics that makes biomembranes so fascinating materials. The most prominent cell membrane shape is that of a human red blood cell. Normally, it is a biconcave discocyte, as you can see in the middle column. However, in different environments, the equilibrium shape is different as well. In high salt concentration, cells shrink to get a spike appearance, whereas in low concentration, they inflate and become almost spherical. So, inspired by the problem to explain the equilibrium shape of red blood cells, in the early 70s, Canham and Helfrich proposed that the equilibrium biomembrane shapes are surfaces of minimal elastic bending energy. This energy, now called the Canham-Helfrich energy, is an integral over the membrane surface M, where H and K are the mean and Gauss curvatures of M, beta and gamma are the bending rigidities or stiffnesses of the membrane, and H0 is the spontaneous curvature parameter. The latter was introduced by Helfrich to represent a possible asymmetry between the bilayer leaflets. and minimization of the Canham-Halfrich energy is constrained to closed surfaces with fixed area and fixed enclosed volume. So here I've written the Canham-Halfrich model again, and just to refresh a little your memories on differential geometry, let me recall that the scalar mean and Gauss curvature can be expressed in terms of the principal curvatures kappa 1 and kappa 2. These are the eigenvalues of the shape operator, which is given by the surface derivative of the unit normal nu. The formulation of the area constraint is straightforward, simply with the Hausdorff measure H2. And the expression given here for the volume constraint follows from the divergence theorem and the fact that M is closed. So the Canham-Halfrich problem is a geometric variational problem because the energy only depends on the shape of the surface and not on its specific parametrization, nor on its position in space or its orientation in space. Of course, in order to have an admissibility class including smooth surfaces, the area and volume constraint data, they must respect isoperimetry, namely that the sphere has the largest volume for a given area. Another important fact is that by Gauss-Bonnet, the integral over the Gauss curvature is 4 pi times 1 minus genus. So therefore the last term in the Canham-Halfrich energy can be omitted when one restricts to surfaces with fixed genus. And so it suffices to consider the energy E0. A famous special case of the Canham-Halfrich energy is the Willmore energy. Its global minimizer is the sphere. And the Willmore energy is well analyzed. However, many existence results take advantage of its conformal invariance, a property that does not hold for the general Canham-Halfrich energy. A necessary condition for minimality, yet alone stationarity of the Canham-Halfrich energy is the Euler-Lagrange equation, which in the context of geometric energies is often called shape equation. And the Lagrange multipliers for the area and volume constraint, they can be interpreted as the surface tension alpha and the pressure difference P. So the shape equation is a fourth order elliptic partial differential equation for the surface parametrization. And this partial differential equation is quasi-linear and therefore difficult to solve. However, there are many numerical studies available, and these studies show solutions similar to observed biomembrane shapes. In particular, with material parameters like this, one obtains shapes that are of the form of the discoid shape of red blood cells. So, but in order to prove existence of minimizers for the Canham-Halfrich problem, the method of choice is the direct method of the calculus of variations. The first task is to identify a suitable function space framework, usually by looking at a priori bounds that can be inferred from the energy. In the case of the Canham-Halfrich energy, one gets the following bound on the second fundamental form of the surface if the rigidities satisfy this inequality. However, this L2 bound is not enough for compactness of smooth parametrizations, which by Sobolev embedding would need LP curvature bounds with P greater than 2. So P equal to 2 is exactly the critical case. And so therefore the direct method does not directly apply in a parametric approach. And the idea is now not to work with a class of parametrizations of M, but with a class of surfaces which has suitable compactness properties. This approach is the so-called ambient approach, and a suitable class of generalized surfaces are varifolds, which I will introduce in the second video. For the moment, let me just note that with varifolds, there exists a counterexample to lower semi-continuity, which is the second key ingredient of the direct method. So one really has to be careful. Nevertheless, in spite of all difficulties, the direct method has been successfully applied to establish existence of Canham-Helfrich minimizers. First, there is the work by Gioxia and Veneroni for axisymmetric surfaces, where the analysis can be formulated in 1D for the rotating curve. Then Dalphin considered C11 surfaces with uniform curvature bounds, and just recently the Canham-Helfrich problem for genus 0 surfaces was solved in the ambient approach by Eichmann and in the parametric approach with generalized immersions by Mondino and Shannon. Let me briefly touch upon another aspect, namely the gradient flow associated to the Canham-Helfrich energy, as I've illustrated here. Generally, a gradient flow describes the evolution towards equilibrium along a trajectory of fastest energy decrease. In the case of the Canham-Helfrich flow, there are now two possibilities to incorporate the constraints, either as globally fixed quantities or as local constraints by penalization of the energy. And the Canham-Helfrich flow, in case of the L2 gradient flow formulation, is this fourth order quasi-linear parabolic PDE, where the velocity vector v points in normal direction nu. This is thanks to the reparametrization invariance of the energy. So analytical results for the Canham-Helfrich flow comprise short-time existence and uniqueness and a lifespan estimate in terms of the initial curvature. Then we know long-time existence if initially the surface is close to a sphere, but otherwise finite time singularities may occur as well. These properties are proved similarly to the according properties of the Wilmot flow, and here I give you some references. Let me also mention that there are powerful numerical models describing the evolution of biomembranes, also including multiple phases, which I will introduce next. So recall the fluid mosaic model for cell membranes, which showed additional lipids and various embedded proteins. The goal is to understand the role of these constituents regarding stable membrane configurations. This role can indeed be very pronounced. In fact, in experiments with giant unilamellar vesicles, one observes heterogeneous membranes similar to this illustration. Here, the different shades of blue represent different phases, and one sees that curvature clearly is coupled to the composition and vice versa. In order to incorporate the effect of such a variable phase density into the Canham-Helfrich model, one has to allow the material parameters to depend on the phase variable, like here beta, H0 and gamma, and one has to include an additional phase energy, and most importantly, one has to minimize the functional with respect to both, with respect to shape and with respect to density. This is due to the lateral fluidity, which allows the membrane molecules to move to a spot which best suits the preferred curvature. Moreover, in addition to the area and volume constraints, due to conservation of mass, also the total phase mass must be fixed. In case of sharp phase interfaces, one does not need a continuous density variable, but one can formulate the energy as a functional dependent on surfaces representing the individual phases, like M1, the dark blue, and M2, the light blue phase. And this multi-phase model was introduced by Jülicher and Lipovsky to describe domain-induced budding. That is, that these darker shaded regions, for example, pinch off. Then one simply has the sum of the individual Canham-Helfrich energies plus a phase energy that penalizes the length of the interface by its line tension. Then the multi-phase variational problem is the following. We have to minimize the energy among compact surfaces, possibly with boundary, such that their union is a closed membrane and they overlap precisely at their boundaries to get the sharp interfaces, and the phase masses and the total enclosed volume are fixed. So also for this problem, there are existence results available. For axisymmetric configurations, there are first the papers by Cioxi, Morandotti and Venderoni, and then by Helmers, where the latter employs a surface diffuse interface approximation. And recently, Elliott and Hatcher presented a surface phase field model for multiphase membranes close to a sphere, and together with Luca Lusardi and Ulisse Stefanelli, my supervisor, we established existence of multiphase minimizers without any restrictions on symmetry. Our settings are varifolds, and in the next video I will introduce this important concept of geometric measure theory and present our result in more details.